This webinar is hosted by a scientist. I think they're in Boston. So, but I think she's from Canada. So listen to the way she says out and about. And if it sounds a little funny, it's because she's Canadian. All right, here we go. Okay, so let me get that out of the way and get that out of the way and start this rolling.
Hello, everyone. So thank you so much for joining us for this mini PCR Bio webinar on COVID-19. I am overwhelmed with the number of you who have joined us today, and we're really looking forward to the opportunity to talk with you about the genetics behind this pandemic that we're all dealing with right now. So I'm Dr. Alex Danis. I'm going to be leading this webinar today, but I'm also joined in the chat by a couple other members of our mini PCR Bio team. So Bruce Bryan and Sebastian Craves are also going to be here, and they're going to help jump in and answer some questions as we go. So I'm so excited to see you all here. And we are a group of science communicators and science educators, and we have a special expertise in genetics. So we're hopefully today going to try and use that expertise to answer many of the really important questions that you sent in before this webinar. But we are not epidemiologists and we are not doctors. So we've also put links to a bunch of different resources, including the CDC and the WHO, in the description down below that you can go to for reliable sources on what's happening with this disease right now and what you can do to help try and prevent it. I also want to point out that this is a very rapidly developing situation. There is a lot of information coming out on a daily basis. There's a lot of science coming out, and it's okay to feel a little overwhelmed by this information. I think even those of us who are scientists and experts feel a little bit overwhelmed by this right now. So we hope that we can empower you with some information and some knowledge so that you can feel like you have a better grasp on some of that that's going on. And we've again linked to some of the best, most reliable resources we can find in the description below and in the additional resources that we're going to talk about at the end of this webinar. So I hope that over the next hour or so, we can help you figure out some of the genetics surrounding the situation and make you feel more empowered to tackle some of the news that you might be hearing about every day. So let's jump in to talking about COVID-19 and some of the genetics behind it. So we're going to talk about a number of different things today. We're going to start off by discussing the basics of viruses, so what they are and how they replicate. We'll then move into answering some questions about what is a zoonotic disease. This is a term you might have heard a little bit about recently, and why do we care about zoonotic diseases? Then we're going to dive into the specifics of COVID-19 and the virus that causes it, SARS-CoV-2. And we're also going to discuss the importance of testing for COVID-19, how it's performed, what biotechnology procedures it takes advantage of, and how it can help us to try and actually stop the spread of this disease. Then we're going to try and take some time to answer some more of the questions that you sent in that didn't fit into the rest of this webinar. And finally, we're going to wrap up with some additional resources for teachers who want to further explore these concepts that we talk about today with your own classes. So I'm excited to have the chance to talk with you over the next hour about all of these things. And again, so many of you are in the chat. I really, really appreciate that you've all decided to join us today, and we want to be a reliable source of information for you. So first up, I want to start with what is a virus? A virus really has two main parts. So first, it has some sort of nucleic acid genome, and this can be DNA like our own genomes, or it can be RNA. So remember that in cells like ours, information is stored as DNA, then it's copied into RNA, and that RNA goes out into our cells and gives the cell the information for how to make a protein. But in many viruses, they actually don't store their information as DNA, they just sort of skip right to that RNA step, and their instructions for making more virus are stored in RNA. The second part of the virus is some sort of shell that will hold that nucleic acid genome. So in many viruses, this is a protein shell called a capsid. And in some viruses, like the novel coronavirus we're going to talk about today, there can actually be an additional shell made of lipids called an envelope. And it's really important that you need both parts of this virus. And again, that's really all there is to a virus. There's a genome and there's some kind of shell that contains it. but you really do need both parts for it to be an infectious particle. So the RNA on its own is just information, and it can't really do much of anything. You really need the genome and that shell around it to actually make it an infectious virus. Now, viruses have evolved to infect a whole range of hosts. You can have viruses that infect bacteria and animals and plants and, of course, humans. And viruses typically, we don't call them living things. So it's a bit of a sort of psychological question of what is life. But typically, we say that viruses are non living because despite having a genome like us, they can't replicate on their own. So one virus on a surface will just stay as one virus. It needs to actually infect a host cell so that it can actually use that host cell's own machinery to make more virus. So on its own, a virus will just remain one virus. It has to infect that host to actually be able to replicate. And that looks a little something like this. So here we're looking at a coronavirus that has these spikes on the outside, and we'll talk about those a little bit more in a second. So these spikes on the outside of the coronavirus bind to receptors on a host cell's cell membrane, and then they fuse their membrane, and the virus is actually able to release its RNA genome into that cell. So here is where it actually takes advantage of the cell's own machinery to make more RNA and more of its capsid and envelope proteins. And so once the new RNA and those new proteins are replicated by that host cell, they can then assemble into new viruses, which can then leave the cell and go on to infect more cells. Now, you may have heard
Use myself. All right, this is our first pause point. First, I need to hear from the teachers. There was a comment in the chat about volume. So if you're a teacher, tell me about volume. And secondly, tell me about the closed caption. Should I turn them off or keep them on?
I like the closed caption. The volume could be a little louder.
Okay.
The video is a little bit quiet compared to your voice.
Okay. Let me see if I can fix that. All right, so you students should now be asking questions about what we've seen so far about what is a virus, and we teachers will monitor that and try to answer your questions the best way we can. Okay, we'll try that. That's not really what I want. There we go. That'll probably help. Okay, I'm going to hope that helps. And there we go. Okay, so I'm looking at the text chat now. Looking at the text chat now. Whoa, what happened there? Chat, 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 chat. Okay. All right. The first question I'm seeing here, is it common for a virus to have the second shell of lipids? So there are different kinds of viruses. And so there are many that don't have this lipid envelope that have instead a protein shell. So viruses are quite diverse, and each is specific to a host organism. If you have seen, can I get a picture? I think I'll stop there. I'm going to be limited in my ability to try things, but the answer to that question is many viruses do have a lipid envelope, many do not. So washing your hands will be effective against some viruses and not. All right, Angelica asks, how does the virus affect the cell's ability to perform its function? Usually, let me go back here and get a nice, okay. This will actually give us a chance to test the volume.
Cell membrane, and then they fuse their membrane, and the virus is actually able to release its RNA genome into that cell. So here is where it actually takes advantage of the cell's own machinery to make more RNA and more of its capsid and envelope proteins. And so once the new RNA and those new proteins are replicated by that host cell, they can then assemble into new viruses.
Okay. To me, that was better volume. Mr. Myers says yes. Okay, good. All right, so the question is, what does this do to the cell? This is really what you care about. You don't care necessarily about the virus life cycle. You care about what does it do to your cells. So usually when a virus does this, it takes over the cellular metabolism machinery and says, make virus, make virus, make virus, and that's all the cell does until it dies. So often this is fatal for the cell that is infected, and that's why you get the disease. All right, let's see, next question. Virus. Okay, I think that's the similar question to what I just answered. Okay, so Luke's question is, how is a virus monitored by scientists? We'll get to more of that in the webinar, so I'm not going to try to answer that now. It'll be better done later. Okay, I think we got some good questions there. There'll be another pause point in about 5 minutes. So let's see what questions you have at that point. Play.
Viruses, which can then leave the cell and go on to infect more cells. Now, you may have heard a lot about something called a zoonotic disease recently as people have been talking about COVID-19. And so these are diseases caused by viruses, fungi, bacteria, and parasites that can be transferred between animals and humans. And according to the CDC, 3 out of every 4 new emerging infectious diseases in people actually first came from animals, which is kind of a crazy number. I was really shocked when I saw that because it's really a large amount. Now, often the viruses or bacteria or whatever it is that cause these diseases, they can exist for a long time in an animal population before they develop some sort of mutation that allows them to begin to infect human cells. So when that happens, you have something that's called a spillover event, where a pathogen at a high prevalence in one population, like animals, comes in contact with another and then spreads to that new population. So this could be a virus at a high population in, say, rats that then comes in contact with humans and has some sort of mutation that now allows it to infect the humans as well. So this happens more and more. We see these outbreaks of zoonotic diseases more and more as we encroach on wild animal populations and we move into their habitats and have more contact with them. We can also get zoonotic diseases from animals like livestock, and so making sure that we're monitoring all of these different populations and trying to keep them healthy is also important in trying to stop these sort of spillover events. And you might be hearing these words a lot lately because scientists think that SARS-CoV-2, the virus that causes COVID-19, likely originally came from a bat population. So to date, the coronaviruses that have caused diseases in humans have had animal origins. So often they begin either in bats or rodents, and there's often an intermediate animal as well. So for SARS, which you may have heard of caused an outbreak back in 2002, the intermediate animal between bats and humans was actually a civet cat. And for MERS, which caused an outbreak that was first spotted in 2012, the in-between animal was a camel. So for SARS-CoV-2, we think that the intermediate animal might have been a pangolin, but we are not sure yet. And so scientists are still working to try and figure this out. And we're going to talk a little bit later about how they're doing that using genetics. So if we can monitor animal populations that we know are at risk of causing spillover events, we can actually try and prevent them before they happen. Or we can try and develop vaccines to combat outbreaks before they become epidemics. So we've been monitoring, so we might be monitoring the original animals like the bats or rats that cause things, we might be monitoring the original animals like bats or rats or we might be monitoring the animals like the pigs that could serve as these intermediate animals. Sorry about that. So this figure right here that we're looking at now shows how different flu seasons in humans came from viruses that traveled between birds, pigs, and humans. And so this figure is actually from a project that used DNA sequencing to look at viral genomes from a bunch of pigs at a very large swine exhibition. And so this project identified a flu virus cluster that looked very different from vaccines that were already available. So what these researchers were able to do was to take this new DNA sequence and send it to the CDC so that they could start developing a vaccine against it just in case an outbreak happened. And so in this way, monitoring the virus while it's still within the animal populations can actually help us to identify viruses that could become a problem before that outbreak happens. So even before the latest outbreak, scientists have been looking at bat populations and sequencing the genomes of viruses that they find to try and get a head start on outbreaks and epidemics because of things like SARS and MERS that happened in the past. And you might be wondering, as I was when I first started hearing about this, why bats? Why are there so many viruses coming from bats? And why are they such big reservoirs of disease? And so scientists have a few theories. One is that their roosting behavior, where they all pack in tightly together to sleep and hang out, actually means that the virus can spread and evolve within the bat populations very quickly. So another key point is that unlike many other mammals, bats can also fly. And so flight requires a lot of energy. So bats have a really high metabolism to keep up with that energy that they need to fly. And it's thought that this might actually help to boost their immune system and keep them from actually getting sick from the viruses that they carry. So it's possible that they're spreading these viruses a lot in these populations because they're packed in very closely. They're not very good at social distancing, and they might actually not be getting sick from those viruses, which really lets those viruses keep spreading. So now I want to dive into
All right, here's our second pause point. So that section was about the origin of the virus. So please ask your questions in the text chat. We'll answer a few of them. Still waiting for a question. Okay, looks like we'll go forward. Minimize that. Start this up again. Okay.
into a little bit of the specifics about COVID-19. And so let's dive into that now. So I want to talk a bit about what its genome looks like, how we can use genetics to watch how COVID-19 is spreading, and also how we can use its genetics to look a little bit at where it might have come from. And one of the other things that I really want to point out is it's important to keep straight what is SARS-CoV-2 and what is COVID-19. So SARS-CoV-2 is the actual virus, and this stands for Severe Acute Respiratory Syndrome Coronavirus 2. And that's a bit of a mouthful, so that's why you can see we've taken sort of the first letters of each of these words and put them together into SARS-CoV-2. And then COVID-19 is the name of the disease that it causes. And again, that just comes from Coronavirus Disease 2019. So it sounds like a lot, but really they're just shortened forms of the virus and the disease. So as we talked about, SARS-CoV-2 is a virus, so it has all of the important parts of a virus that we talked about before. So it has its own genetic material, and in SARS-CoV-2, this is RNA. It's an RNA virus. And that RNA is wrapped in nucleocapsid proteins. So here in this figure, you might be able to see these little proteins that are wrapping this RNA. So these are the nucleocapsid proteins. Also around that, it's surrounded by a lipid envelope. And so embedded in that lipid envelope are spike proteins. And you've probably seen these in a lot of pictures that are popping up on the news. The main image from the CDC colors them as red, so it's got these big red spikes on the outside of the virus. And those spikes are why SARS-CoV-2 is actually classified as a coronavirus, because these viruses are covered in these spikes, and when you look at them under a microscope in certain ways, it actually looks a bit like a crown or a corona. And so the lipid envelope is also really important to us for trying to keep ourselves safe from SARS-CoV-2 because good old soap can disrupt it. So washing your hands is truly one of the best things that you can do to try and keep yourself and others safe from getting COVID-19 because the soap molecules can actually go in and disrupt that lipid membrane. And as I mentioned before, if it's just the RNA left behind, it's not infectious. So you may have heard different studies talking about the fact that people have found SARS-CoV-2 RNA on a surface 17 days after the last person was there. But if it's just that RNA, it's not really an infectious particle. The RNA alone is not infectious. You need both parts. And so if you can wash with soap and you can disrupt that lipid membrane, you're not only washing the virus off, but you're also sort of breaking it open. Now, that RNA genome of SARS-CoV-2 isn't very big, so really the virus's RNA genome doesn't need to contain too much information. It just needs the information to be able to make more virus. So it's only about 30,000 bases long. And if you think about our own human genomes, those are 3.2 billion bases long. So its genome is really relatively very small. So it encodes for a few key proteins, and I've labeled just some of them here. So there's a big open reading frame, and what this is it's just a section of the genome that has all of the molecular signals needed to be able to make a protein. And then when that's transcribed by the host cell, that will actually be turned into proteins that will help to replicate the viral RNA. There's also a gene that encodes for those spike proteins, and that's what binds to those receptors and actually lets it into the cell. And then there are also a number of proteins there, genes that encode for proteins that are involved in making that capsid and then that envelope on the outside. So doesn't have a ton of information, but it doesn't need a ton of information. Now, RNA.
Okay, so that was discussing some of the viral structure and viral genes. Once again, we're pausing to see if there are some student questions. Okay, we got a lot of questions now. All right. I'm going back here. Okay. All right, let's take Amar's question first. Could COVID-19 mutate in humans to become more contagious? Viruses are continually mutating. You can see the slide that I have on screen right now is going to talk about how quickly does this particular virus mutate. But they are always changing, and that's why we had this disease that didn't exist 2 years ago, because the virus that caused it didn't exist 2 years ago. So the specific question is, could it mutate to become more contagious? The answer is certainly it could. Okay. Kyle, we need your question to be more specific. When you say methods of infection, I don't know exactly what you mean by that. And Lucas' question is, how long does it normally take for an animal virus to be able to infect humans? It's going to be statistical randomness. There was something I watched yesterday that talked about this is the third coronavirus to really affect humans in a scary way. The first one was SARS 2002. Coronaviruses as a group have been around for thousands of years. So normally when they affect humans, these coronaviruses, they make you sniffle and sneeze and you say, okay, I'm better the next day or 2 days later. So the common cold can be caused by a coronavirus. But we're seeing these scary ones. This is the third. And the title of the talk was Third Time's the Charm, because from a virus perspective, it is. This is from the virus perspective, this is glorious. They're infecting millions of people, whereas the first two only affected a few thousand people. But the fact that we had 3 new scary coronaviruses in 20 years suggests that the mutation rate and the passage from animals to humans is becoming much, much more prevalent than it has been in human history. So something to worry about. Okay. All right. Okay, Kyle has a question about survival. Survival of the virus, not survival of the humans, survival of the virus. I have that data. I will show it tomorrow because there's a paper. There's a famous paper that did the study of how does it survive in the air and how does it survive on a plastic surface and on a copper surface and then cardboard. I don't know why they chose copper, but they did. Stainless steel, I understand, but what's made of copper in our world? Pennies. So I'll save your question for tomorrow, Kyle, but it's a great question. How long does the virus survive in the environment, right? Okay, beautiful. I think, okay. Luke Johnson has a question about high metabolism of the bats being important for how it spreads. There's not a lot of information on that, so that's really speculative at this point. Bats are weird, and the fact that so many of these modern viruses are coming from bats is a puzzlement, and people are guessing. Okay.
Can I add to that really quick?
Sure.
I think what she was saying was that they carry these viruses, but because they have a high metabolism, they're not affected by the viruses.
Yeah, but the one of the weird things about bats is they have no bone marrow. It's too heavy to fly, which means they have no B cells, which is it's half our immune system. The immune system is B cells, which make antibodies. So bats don't have those. So that could also be a factor. So bats are weird. All right, going back to the webinar. This next section you can see is going to talk about virus mutation. Very important in the virus's ability to survive. If it did not mutate, it would not be able to survive very long in the environment. Well, in living creatures, right? In living creatures. Here we go.
RNA viruses like SARS-CoV-2 often have really high mutation rates. So this might be something you're hearing about in the news, is the mutation rate of this virus. So in our human cells, when our DNA is replicated, we have a kind of proofreading mechanism that goes through and fixes any mistakes that are made as our genome is replicated, as those strands are copied. But RNA viruses don't have this, so they tend to accumulate lots of small little changes and mutations. And this sounds like it would probably be a bad thing for the virus, but it's actually an advantage for them in most cases because it allows them to change and evolve rapidly to try and evade the immune system of their host. Now, the mutation rate of SARS-CoV-2 is about 2 to 4 times slower than the seasonal flu virus. And so scientists are seeing that it mutates at a rate of about 2 mutations per month. And there's a great article that will be linked in the resources as well that talks about this almost like a big game of telephone. So if I have the information for that virus and I'm going to spread it to 2 people, it's like me telling that genome to those 2 people. Now they're going to go and they're going to tell it to 2 people and they're going to tell it to 2 people. And if you've ever played a game of telephone, you know that mistakes happen. And so as those mistakes happen through the population, you can actually track how often they happen. So with the mutation rate of SARS-CoV-2, it would mutate at a rate of about 2 mutations per month, which would be a small mistake occurring every second or third transmission. So in that game of telephone, every second or third person would make a mistake in that message that they were relaying. But I want to also note here that the word mutation sounds kind of scary, but I don't want you to be scared of that word mutation. Most of the mutations that are happening in this virus don't actually affect its function at all. Rather, each mutation, or because each mutation on its own is not likely to have a really big effect on how infectious it is or how severe it is. But instead, these mutations actually let us do useful things like track the virus as it moves through a population. So I want to do, I want to talk about that a little bit with a sort of example here where instead of the viral genome being a long strand of A's, C's, U's, and G's, that big long viral RNA, instead I want to imagine that the viral genome is this sentence: the cat and the rat are mad. Now, as you're playing that big game of telephone and you're passing it on to people, at some point there might be a mistake made where now there are two different messages going out. One that says the cat and the rat are mad, and one that says the cat and the bat are mad. Now, these keep propagating through the population, and maybe another mistake happens where now you have one that says the cat and the bat are sad, as well as the cat and the bat are mad. So you can look and see how these different, in this tree, you can see how these different mutations happened and sort of what that process was. But we're not actually able to sequence the viral genome in every person it infects as it infects them. So instead, when we sequence the virus, when we look at that viral message from all of these different people that we've taken a sample from, we just see this. We see three things. We see the cat and the rat are mad, the cat and the bat are mad, and the cat and the bat are sad. And as scientists, we have to figure out which was more likely. Was it more likely that the sequence of events was what's on the top here, which says the cat and the rat, then cat and bat, and then bat and sad? Or is it more likely that it went cat and rat, then bat sad, and then back to mad, but now also still bat? And so this here, it's really about a scientific concept called parsimony, which is a very fancy word to say choosing the simplest scientific explanation that fits the evidence. So it is far more likely, I think, if we all look at this, that the top version, this sort of 1, 2, 3, is much more likely than the bottom version. So we can then use that to actually go back and try and recreate this sequence of events. So when we do that with the actual viral genome, we get something that looks like this. So this is a phylogenetic tree, and it's really just a much more filled in version of the dummy one that I just showed you before. So just like the fake tree we just looked at, phylogenetic trees are all about building the most likely series of events. So we've sequenced, or scientists have sequenced, the virus's genome from people all around the world, and they're trying to look at how those mutations likely happened one after the other. And genetic sequences are a really great source of evidence to try and build these trees, though we can also use things like location and timing to try and add additional evidence to build the most correct tree possible, or our best hypothesis tree possible. So it's kind of like a family tree where you started off with one virus and then now you have sort of that virus's children. So you can think about this like a family tree where the left is sort of the oldest virus and the right is the youngest virus. So we're moving from left to right through time. Making sure that's the right way on your screen, but we're moving from left to right through time. Now, another thing I want to note here is that these can be very, very useful to trying to track this mutation through time. But we're not sequencing the virus in every person as it's affecting every person. So this is an incomplete tree just by its nature, because it's just not realistic for us to be immediately sequencing every person as they're being infected and passing it on. But it is the best interpretation that we can make right now with the information that we have available. So it's a useful tool for scientists to try and track how this virus is spreading and moving, but additional sequencing can help to change some of those branches and could change how we believe viruses have spread. It's just about getting as much evidence as possible to build the most complete trees that we can.
All right, here's our next pause spot. We'll monitor the chat to see what questions there are. Okay, Kian has a great question. What is the purpose of tracking mutations in terms of helping people treat COVID? The specific question of treatment will not rely on the mutations. This is a bigger picture investigation. Where did it come from? How likely is it to change in the future? If we make a vaccine for it this year, do we have to make another one next year? How quickly does it change? With flu, you need a different flu shot every year because the strains have changed in the intervening 12 months. With this virus, nobody knows yet. So understanding how fast does it mutate will affect the prognosis for how long is a vaccine good for, or if you get it once, can you get it again? If it's mutated in between, the answer is yeah, maybe you can get it again. All right. Okay, so I'm going to go forward. You'll also see that the next section is just going to talk about is the spread among humans, which can also be partly traced by using this genetic data, the mutation data. Okay, going to go forward. I think this is the last section, and then I want you to tell me questions that you would like to see discussed tomorrow. All right, let's go here.
And track how this virus is spreading and moving, but additional sequencing can help to change some of those branches and could change how we believe viruses have spread. It's just about getting as much evidence as possible to build the most complete trees that we can. So you can also look at those phylogenetic trees like this. And so this is sort of trying to look at how this virus might have spread around the world based on that tree. And I don't want you to try and interpret this because it kind of looks like a giant hairball because there are so many connections here. But scientists can try and interpret this. We just can't do it straight from this picture right now. But I think what I want you to get from this is that there are a lot of connections here because we live at a time where we are just exceptionally interconnected around the globe. And that is wonderful for a lot of reasons. That's a really great thing. But it also means that these diseases can spread pretty quickly too. So it's one of the reasons why we're very exposed to pandemics like this at the moment. But doing this and being able to track these and look at them, I think, is really powerful for scientists as we try and watch how they move around the globe. Now, these mutations in the genetic sequence not only let us track how the virus is now spreading, but they will also let us look backwards and try and figure out where the virus came from. So this is a great paper that dives into this called The Proximal Origin of SARS-CoV-2. And what it did is it looked at the viral genome that is infecting humans, but also at the viral genomes of viruses present in other animals like bats and pangolins that we've talked about a little bit. And so again, we're trying to trace back to see the most likely series of events for where the virus came from. And so the virus that's currently infecting humans is very similar to a virus found in bats right now. So as we look at those similarities, it's pretty clear in the data that the likely origin, the original origin of this virus was in bats. Now, the data also suggests that it might have possibly moved through pangolins, but we're still trying to figure that out. We need to do more sequencing. We need to look at other animals. We need to figure out what is the most likely sequence of events based on that genome sequence to get to the virus that we have today. Now, we did receive some questions before this webinar about whether or not the virus had actually potentially been designed by humans and originated in a lab. But studies like these are exactly why we know that it is far, far, far more likely that it actually started in a bat, because we can really clearly trace where it came from and how it's getting to us now by doing this kind of DNA sequencing. So this paper dives into that a lot more as well. I don't want to spend too much time on it today, but doing this kind of DNA sequencing can let us trace it back to those bats rather than another source. Now, the other thing that it lets us do when we're looking back in time is because we know the mutation rate of how many mutations this virus accumulates in a given month, it also lets us look back and figure out when it actually emerged in humans. And so we can back calculate and figure out the fact that this disease started in late November and December and not earlier. So that's a lot of information about DNA tracking of this virus and how we can use these sequences to look at populations. But the other thing we want to talk about is what makes SARS-CoV-2 unique. Why is this really causing this pandemic that we're seeing today? And there are a couple of different things. Um, so, as we've discussed before, zoonotic disease outbreaks do happen. So, we've seen them already in 2002 with SARS and then 2012 with MERS. Um, but this outbreak is actually far more reaching than those because it binds to the ACE2 receptor in a new way. So, it's created this new spike protein that can bind to this receptor in a new way. And also, the way that it's spreading through populations, it's more infectious than things we've seen before, like the flu. So you might have heard of a term called R naught. It looks written out like R0, but it's pronounced R naught. And this is a measure of if I have the disease, how many people am I likely to spread that disease to? Now, again, because we're really in the middle of this outbreak at the moment, there is sort of a range of estimates for what that might be between 1.5 to 3.5. So that means that if I had the virus, I might infect 1.5 to 3.5 people if I wasn't social distancing and I was just going out there into the world, which is a bit higher than the flu. Which is why it's spreading very quickly when we think about it in comparison to things like the seasonal flu. But it's actually much lower than things like measles, where that R naught is somewhere around 12 to 18. So again, scientists are studying this at the moment. This is a bit more of an epidemiology thing. And again, the CDC and the WHO have great resources on that linked below. But it is spreading through the population pretty quickly. So how can we actually try and contain it? So hopefully you have all been washing your hands and social distancing as much as possible. But another really key thing is testing and isolation. So we want to be able to test people in the population to figure out, do they have the SARS-CoV-2 virus? And if they do, we want to isolate them from other people so that they can't pass it on. So here we have a little example here. Each of my purple circles is a person, and each person is going to spread this virus to 2 other people. But if we can test those people and know that they have the virus before they actually pass it on, we can isolate them and really dramatically reduce the number of people who end up being affected at the end. So how can we actually test for COVID-19 and this virus, SARS-CoV?
Okay, this is our last pause point for today. You can see this is the second half of the webinar, is how do we test, and I think at this point with our time that we'll save that for tomorrow. What I would like you to do now is think carefully, what questions would you like to know, would you like to have answered about this disease? They don't have to be related to the webinar we've seen. All of us teachers will take note of these questions. We may be able to answer a couple today, but some of them will be better done tomorrow when we can actually pull up the graphics that will help you. Words are one thing, pictures are better. All right, so I'm going to look at the chat and see what we've got. Okay, we're starting to get some good questions now. One of them is talking about mutation. Can the virus be mutated enough to stop infecting humans? And the second question is, what part of the virus actually harms people? So the biochemistry of that. What is the chemical makeup of the treatments? That's actually a great thing to talk about as well. That's a lot of chemistry. All right, of those 3, the one I could answer today is, can the virus mutate enough to stop infecting humans? The answer is yes, but usually, if you remember the branching tree, Let's just go back. This is a different kind of branching tree. Let me go back to this branching tree. This branching tree. All right, this branching tree is the idea that you start with, let's say, one virus, then it becomes two. It's not like the mutation eliminates one of them. It's that now you have two different ones. Each one can go about doing its thing. Maybe this one infects humans here. This one infects pigs here. The human virus doesn't cease to exist because you made a pig variant. It's just now spreading to more places. So the virus will certainly can mutate so it no longer affects humans, but that doesn't eliminate the one that does infect humans. So that's the usual way of genetic diversity. All right. The other questions about the chemistry I'll save till tomorrow. Let's do this. Let me make a decision here. Let me look at my total participants, Participants. All right, we've got some more questions. Let's see what these lead us to. All right, yeah, so there's a question about the social distancing. Where did that 6 foot number come from? That's related to what Kyle was asking about earlier, which is great. We should talk about that. We should talk about that. I'll put it on the agenda for tomorrow. And question about why is this outbreak worse than the other SARS variants? That's a good question as well. All right. Okay, I'm going to I'm going to make some breakout rooms, and that'll allow you to speak out loud. Often speaking out loud is easier than writing something in text. And when you hear somebody say, oh yeah, good question, I wonder about that too. I'm going to make some breakout rooms and send people to them. Breakout rooms. Okay, we have 4 teachers, I believe, so I'll have 4 rooms. I will assign you manually. And breakout room 2 will be Mr. Myers. We'll put, okay, I'm putting Mr. Myers' students there. Okay, got a lot of Mr. Myers students in a room. Excellent, excellent, excellent. Okay, Will Coffee, you're the last on the list. All right, room 3. Actually, let me do room 1. It's mine. And doom, 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 doom. Doom, 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 doom. Okay, so this is going to be a mixture of ACR students and chemistry students. Go into room 1. Doot, 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 doot. Okay, go into room 3 now. This will be Mr. Duong's students. Okay. Alex Wyman, is that yours, Mr. Duong? Or is that Parda's? I'm going to put you all in the same room. Then I don't have to guess. Okay, boom. All right, here we are, manage participants. Okay, I am unmuting all. So if you don't want us to hear you, you can mute yourself.
I'm Mr. Sogo.
Hello, Joey. All right, Amar's got a nice icon. That's a T-Rex, huh?
Yeah.
Excellent.
So
Hi, Mr. Sogo. Sorry.
Hello, Anna.
Go ahead.
I'm not sure who that is, but hello, random citizen. Maybe it's Cole. Yes. Okay, good. Good. All right. I think you were saying something, Amar?
yeah, I had that previous question in the chat about why the other SARS variants were less serious than this one. Is there a
Yeah.
particular reason for that? Or?
I know the numbers, and it's really a completely different game. But I need to see. You're talking about a mechanism, which is great. I don't know that. I will try to find out before tomorrow. All right, Harley.
Hello.
Anything? Hello, Harley. How you doing?
Good. I don't really have any questions right now.
Okay. Does that mean that you understood things well and you're satisfied, or that you say I don't know what to ask because I don't understand anything?
I understood what things well, and I'm kind of like thinking about it, letting it boil. I might have to ask tomorrow.
Yeah, exactly. I think that's a good way of looking at things. Often that you don't know what you want to ask if we don't give you enough time. All right, so maybe you'll come back tomorrow with some good questions. That's perfect. All right, Josiah, good.
Yeah, I'm all good.
Okay,
Mr. Sogo, I have a question.
Nathan, go ahead.
So I read on the news that there's some tests showing that using malaria treatment disease medication, like I think it's called chloroquine phosphate, as a potential use for curing the coronavirus. And I was wondering, is that just scant evidence? Is that just like is that like a skeptical hypothesis to make? Or use medication for like a different disease?
Yeah, so that's a great question. I don't know. I don't have the data, but typically in a situation like this, to actually prove something is effective, you need quite a bit of time, and so we haven't had enough time for anybody scientifically to get that data. So I will find the data and I'll share it with you tomorrow. I'll share what I can find.
Okay.
So it's a good question. Absolutely. Yeah. The idea of what are the treatments, and people are saying we're testing vaccines now, and so yeah, what are we testing? That's great. Hello, Luca.
Hi, Mr. Sogo. So in the video, she mentioned that the mutation rate was like 2 per month.
Yeah.
Is that high relative to other viruses?
I am not used to hearing it in terms of per month, because normally you express it as in terms of generations, and so I don't know the answer to your question, but I'll try to find out more. Yeah, because you're right. That's an interesting thing to wonder about. It is lower than influenza, which is flu, and so flu is highly mutable. This is less so. Okay. Good question. Thank you.
Mr. Sogo.
Yes, Harley.
So when trying to develop a vaccine for corona, or like trying to use like the medicine for malaria, like what are they trying to do, like to quote, like fix or stop or cure corona? Like how?
Excellent. Yeah, excellent question. So clearly that's a question on many people's minds. So what I'll do is I'll get the resources so I can do that well, because I need to show you the pictures. And that may be the most important thing to do tomorrow, is to say what actually are these molecules within the virus, because it's the biochemistry, and that's what biochemistry is. It's the molecules that allow things to happen in your cells and in this virus. So I'll try to get all those pictures together and be able to talk about that intelligently.
Thank you.
Thank you.
I had a question on how the virus attacks the body. There's been a lot of it says to be mainly attack the the tissue in the lungs and
Yes.
like stuff like that. But why specifically in the lungs? Is that something in the RNA that it makes more susceptible for the lungs to attack that specific type of tissue?
Okay, great. Yeah, so that's another molecular mechanism, biochemistry. Okay, so I from your questions, thank you all for asking these questions. I have a clear view on what appears to be the most interesting topic for tomorrow. And so it may be, I was originally planning to use the second half of this mini PCR, but maybe I'll cut that down to just like 5 minutes and show the most important 5 minutes and then talk about what is the biochemistry of this virus and what are the molecules that humans are thinking about using to fight it. And all that data will be certainly interesting for me, and I hope interesting for you as well. All right, I am going to leave this room. I'm not sure whether Ms. Holtz also left this room. So you're welcome to talk to each other and just say hi. And it looks like there's a lot of ACR people here and a lot of non-ACR people here. I'm going to join another breakout room and just talk to the people there. If you want to hang up and leave, just say bye-bye. That's great. I'll be back to this room in about 3 minutes, I think.
Bye, Mr. Sogo. Thank you.
Bye-bye, Joey.
Bye.
You. Bye.